Now I have the pleasure of introducing our first lightning speaker of the day, Dr. Marianna Rapsomaniki.
Perfect, thank you.
She is a staff research scientist and a group leader of the AI for the single cell research team at IBM Research, Zurich. Her talk today on precision medicine in the age of AI and quantum computing is extremely interesting, and I'm eagerly looking forward to it, as I'm sure many of you are. The stage is now yours, Marianna.
Thank you. Can everyone hear me? Okay. So thank you for the invitation. Thank you for the excellent organization so far. We're going to stay on the topic of health, but we're going to dive a little bit more deeper in what happens in our cells, right? So, and let me first explain a little bit about what has been the latest big revolution in computation biology, in biology in general, which is the fact that we are now able, for the first time, to quantify a series of different macromolecules, DNA, mRNA, proteins, and so on, with single-cell resolution, so at each individual cell. And we can do it for different molecules at the same time, so we can have multimodal data. We can do it while preserving the topology of the tissue, so in space, and over time. And all this is telling us that very, very soon we are able to profile each individual cell of a whole organ, or even a whole organism, as you see here in this plot of all human cells from different tissues, down to the last single cell for millions of cells, maybe even trillions of cells soon. And we can do it in health, in disease, over time, and so on. So as you can imagine, this has been a big revolution in biology, computational biology. This has enabled us to train really big models using these data sets. And it very importantly allows us to go from what we call like a protein target-centric approach, so identifying unique molecules, unique biomarkers, and trying to inhibit those with drugs, to a cell-centric approach, which is what you see with this plot over there, which is a three-dimensional reconstruction of a breast carcinoma. So we're now able to understand how these cells exist together in a community, and if we're able to do that, then the hope and the vision is that we can try to reverse disease at the cellular level, where it actually occurs in its native environment. So this is what we try to do in the team, and we've been doing this for quite some time now. One landmark study that we were a part of was published in 2019, and there's a very nice special from Reuters, if you're interested in a small summary of that, where what we did is analyze a big cohort of breast cancer patients, and then we analyzed all individual cells in these tumors, both cancer cells, but also very importantly, immune cells or other cells in this environment. We try to understand these interactions between all these different cell types. And what this study showed is that it was able to point to some new insights about how tumors are organized, how this heterogeneity is important for the progression of the disease. And it also allowed us to design better precision medicine approaches. So this was all great, but there was one thing missing, which was the fact that in this study, we had to dissociate the tumors because of technical limitations of 2019. So we could not understand how the cells are in the tissue, what is next to what. But things have changed since then, so now we can actually do that. There are experimental methods where we can do that. And these methods have shown what biologists always knew, that tumors are specially organized heterogeneous ecosystems. And this means that you have in these ecosystems a lot of cancer cells, but also a lot of immune cells, stromal cells, and so on. And how they interact is very, very important for what happens to the patient. So the spatial organization is very important. And what I'm showing you here is a very famous plot for any biologist. Anyone with a biological background definitely has seen it, one of the most highly cited papers of all times, which is the so-called hallmarks of cancer. So what these are is emerging properties of tumors that are very predictive of certain disease outcomes, like the role of the immune system, the immortality of the cancer cells, and so on. And what we now know is that all these interactions are related in space, are related to a lot of these hallmarks. They give rise to different hallmarks. And that's exactly what we try to understand. So we try to understand interactions between different tumor clones themselves, whether antagonistic or synergistic. We try to understand, very importantly, immune tumor interactions, Because what happens is that ideally immune cells would target and kill the tumor cells, but very often what happens is that tumor cells inhibit and trick the immune cells to evade this getting killed by them. So this is what we want to understand. But also the role, for example, of the blood flow to the tumor that gives different nutrients to the tumor, and so on. So all these different spatial characteristics we can now profile and quantify with single-cell resolution. And what is very important to understand is also the role of different treatments, right? Because one of the leading causes of cancer-related deaths is the fact that when you give a different drug, this kills different populations, but if certain populations manage to survive in the tumor, this can be very detrimental. They can develop resistance, and this is what actually kills tumor patients. So this is the focus of the team. This is what we're trying to achieve. We use a lot of all these new, very, very exciting modalities, multimodal data, and so on, and we exploit with different AI and machine learning techniques with the ultimate goal to enable AI-driven precision oncology, as we say. And why this is important again is because if we are able to design these microenvironment-informed treatments, as we say, we can design better immunotherapies if we understand this immune-tumor interaction. We can design immunotherapies that are appropriate for given solid tumors so that the immune cells can actually access the tumor cells, but also understand which patients will profit from immunotherapy, which is a very big limitation today. We can understand how resistance happens, how cells acquire resistance, or if cells have pre-existing resistance, target those and kill those more effectively. So how we do it with AI. So a big part of the research that we do in the team has to do with representing these ecosystems. Data representation, of course, is very, very important. And the most natural way to represent them is using graphs. And in these graphs, you would have nodes representing cells, edges representing connections between the cells, and for each node you would have an annotation that would tell you what proteins are expressed, what genes are expressed, and so on at the cellular level. So what you're trying to understand is this social network of cells, in other words. And it's, of course, many of you probably already guessed it, that we use a lot of graph representation learning for that reason. So real tumors really look like this. This is just small snapshots of real tumors. They can be very, very different. So these graphs can have very different properties. Some are more modular, some are more heterogeneous, more sparse, and so on. And this is the information we're trying to ingest and use for inference. So how we do it is by taking this data, segmenting these images, which is already a very big problem on its own, where AI has helped a lot, identifying, clustering this data, identifying these different populations, generating these graphs using different assumptions of how cells communicate. And one first approach that we tried in the team is using these graphs, extract handcrafted features from these graphs that are expressing, for example, how infiltrated a tumor is or how heterogeneous a tumor is. And this is the work of Adriano Martinelli, our PhD student in the team, that was just published last year. And what Adriano did is to develop a computational framework, you can find it on our GitHub, that is doing exactly that, is taking this data, ingesting this data, generating the graphs, and then computing these different features, which we can use for downstream analysis to understand, for example, if we have a patient cohort, why different patients responded different to immunotherapy, let's say. And this is what I'm showing you here, like a quick example. Starting with these images, what you would have is, let's say, staining of this tissue with different proteins. So you would see what proteins the different cells express. You can use this information to identify, is it an immune cell, is it a cancer cell, what type of immune cell, what type of cancer cell, so you can really have a very detailed look into the identity of each single cell. Ultimately, constructing these graphs, different assumptions can be used here of how these cells communicate in space. And what you do in the end is compute these scores that I mentioned. So what I'm showing you here up left is, for example, something like an activity map, like we call it a local entropy map, that just highlights the regions that are very rich in interactions between very different cell types. Or below that, you see regions of the tumor that are very infiltrated. Or with a heat map, you see how these different cell populations interact. Do they attract each other, which is shown in red, or do they avoid each other? So all these types of things we can compute right now with these handcrafted features. But what is also very exciting is like taking this a step further, and instead of extracting these scores, try to learn it directly from the data using a graph representation learning approach. And what we are trying to do now as a next step is use these graphs, identify subgraphs where all these different hallmarks are active. Like, for example, maybe some cells are responsible for evasion. Then we have a subgraph that describes evasion. Maybe some others are responsible for tumor-immune interaction. We have a subgraph for that. And then, which is the work of Santiago, what we're doing is using these hallmark graphs in a graph representation learning scheme, concatenating these hallmarks and trying to predict some patient metadata. And this is something that is underway right now, but it's a project we're very excited in the team. And the goal of that would be to identify which are the exact interactions in the initial tumor between different cell types, which can be seen as a spatial biomarker, if you want, so a pattern of interaction, and then go back to the patients and say, okay, how often do different patients have these types of interactions? Is this predictive of their outcomes, their survival, and so on? Because if you know what this interaction is, then you can actually target it, you can inhibit it. So moving on, as I told you, we work a lot with data. Multimodal data is very important here, of course, and this multimodality causes a lot of issues, technical issues that we need to overcome, how to integrate the data, how to predict one modality from the other, what happens if for half of the patients you're missing one modality, and so on. So we work a lot on that, and I'm showing you some two very recent works. One is called MatchClot, and is the work of Federico, where we ask the question, can we use these different modalities? What you see here is just two different ways, two different types of single-cell data. One is telling you how accessible the chromatin is, and the other one is telling you the gene expression, how much is each gene expressed. So can we find an algorithm, can we devise an algorithm that matches these two different modalities? And if you make an example like speech to image, for example, if I have an image of a dog chasing a cat, and I have a string that tells me the dog chases the cat, can I match these two? Okay, these two representations of the same thing. And if you can do that, what is very interesting is that then you can view cellular function from different angles, and you can understand it much, much better. So what Federico did is to devise an algorithm called MatchClot, where, without going into a lot of details, it's inspired by contrastive learning, recent developments in contrastive learning and optimal transport, and is doing exactly that. And I'm showing you, just going straight to the results. So what we did first is to try it on a public benchmarking data set from a NeurIPS competition, and we were very happy to see that it outperformed the top scoring methods. But what was very interesting is that we could plot these different data representations using the real matching between the cells, so the real correspondence of what comes from the same cell. We get an embedding, which is what you see on the left, and when we did it with the algorithm, we got almost an identical embedding. Of course, it's flipped because of how these embedding algorithms work, but it fully preserves the structure of the data. So this tells us now that we can go and take any data that is not paired and pair it artificially so that we can fully capture the identity and the function of these cells. And what was also very exciting to see is that even trying this in a zero-shot fashion, in an unseen data set from completely different origin, we also performed a little bit more noisy embeddings for technical reasons, which is expected, but still we performed very, very well. So the work is, there's a preprint out, and it was just accepted in a journal that you see the citation there, but it's also on our GitHub. Okay, and moving on, I'm very, very happy to also work with very talented people, and one is Dr. Pushpak Patti, who is a postdoc in our team, and what Pushpak did is that he got very, very inspired by recent developments in image-to-image translation. And I'm sure you've all played, maybe occasionally, putting your photo somewhere on a website and then generating it in different styles, right? Medieval you, or I don't know, with different artists, different painter styles and so on. So what Pushpak thought is, why don't we apply the same algorithms to images of tissues, of biopsies? And the thing that happens there is that there are many technologies that we are using to profile these biopsies. And the cheapest one is the pink one that you see there. This is called H&E, but we also have others that are much more expensive and difficult to generate. The problem is that we cannot generate it from the exact same tissue, however, because we cannot stain it twice. So what Pushpak did is that he took this source image, which is very cheap to profile, and this one had different stainings of different proteins using a technology called immunohistochemistry. And then he devised a method that on the content of the first image transfers the stain of the different stainings of the different proteins, which is what you see in the bottom. So you can already see the bottom row has the exact same architecture content as the first row, but these are all fake images. Right? So then the question is, can we trick people that these are fake images? And it looks like we can. But what is very important is that this allows us to generate paired data by transferring the staining from the real one to the content of the source one. And very, very quickly, before I leave you, just to tell you that a big project that we just started in the team is called Prometex. It's a very interesting collaboration with the University of Bern and EMBL, and we're trying to use these methods using patient-derived organoids profiled with different drugs to devise an atlas of different states. And the aim here is having an initial population of cells. Can we predict, if we give a drug, where these cells will shift in the metabolic space, in the phenotypic space, and can this tell us which is the best treatment? And the last teaser is that working at IBM, of course, I'm very privileged to work with a lot of people that are very excited and are very experts in quantum computing. So we thought, okay, what if quantum computing is the new frontier? Because it is allowing us to do computing in a radically different way. So what we are currently exploring now in the team, in collaboration with a lot of colleagues from the IBM quantum team, is how can we use different types of embeddings in a quantum computer to learn these tumor representations more efficiently, and how can we predict these drug perturbation models, again, more efficiently. And with that, we are a happy bunch of people that you see over there in the AI for Scientific Discovery group. Please check our career pages if you're interested in all topics AI for the sciences. A bunch of people contributed to this work that I mentioned throughout the slides, but also very exciting collaborators, and I'm looking forward to the questions. Thank you.
Thank you very much, Marianna. So I'll kick it off with a question from Slido. Mm-hmm. And you sort of addressed some of this, but the one question that comes up here, do you have models that learn incrementally?
Oh, that's a very good question. So there is some work from some people in the team, how they do incremental learning from tissue biopsies. We don't have something right now, but it's definitely a very interesting research direction, because data would keep coming in. So yeah, there is some work that I can point you to that is very, very related to that, but not from the things that I showed you already.
Thank you. I'll open it up to a question from the audience, if there's any. A couple in the front here.
Hello, nice talk. I have a lot of questions because we work in the same field, but to start with, I wanted to know if you used any algorithms for studying tumor progression, because I didn't see any dynamical time models.
Yeah, so for modeling tumor progression, you would need to have longitudinal tumor data, right? And the problem with that is that you cannot cut open a person and take a biopsy because you want to do a study, right? So we have some data that is longitudinal in the sense that we have metadata because of patient follow-up, but not the exact same tumor over time. What we're trying to establish now with collaborators is a way to culture these tumors in a dish, in an ex vivo way, for, let's say, 10 days. But then the idea is that you take, of course, after surgery, you take a slice of the tumor, you embed it in a medium, you culture it. Maybe you can put a drug, you can see what happens. And this would be a way to see how the tumor grows over time, but not in the patient, right? And then there are approaches that are more simulation-based type of approaches where you simulate in a completely, let's say, you start with certain assumptions of how cells grow and divide and so on. But what we are trying to do is a little bit orthogonal to that because it's more of a data-driven learning. And since it's hard to get this data, no, but maybe we should talk, maybe you have good ideas about that.
Yes, thank you. So I may ask a second question? If so I was wondering about the effects of the errors, basically, in your image segmentation on the
that's
on the cell-cell communication that you infer at the end.
That's a very good question, because everyone in the field is struggling with that. There are some very recent works from people that trained really big models to do segmentation for different types of cells which we're using. And they perform very well, reasonably well, but the problem is that for certain cell types, like fibroblasts, it's almost impossible to segment them. Even people cannot even experts cannot segment them. So the answer to that is that segmentation performance, what we do is we take the models, we apply it on the data, we fine-tune the models, we use ground truth from experimentalists to go back and fine-tune. But I think we need to accept that for certain cell types, this is always going to be suboptimal. And if we know it and we don't try to make too much, inference based on that, I think it's good, but it's a limitation that everyone has.
Yeah, exactly. Thank you. That was my point, because indeed there are so much errors for some certain cell types.
Absolutely.
And, I would say lack of actual ground truth.
Yeah, so it's impossible. I mean, we were showing the images to the experimentalists and asking them, can you draw like the mask of where is the cell? And they're like, we don't know. They can only see the nucleus with high precision, but the whole cell is very hard.
And even the binucleus cells will be even...
Yeah.
Thank you very much for all your questions. Thank you very much, Marianna, for your excellent talk.
Thank you.